We're joined by Dr. Megan Baldwin, who is the CEO and Managing Director of Opthea. Opthea is developing OPT-302, which is a novel biologic inhibitor of VEGF-C and VEGF-D as a complementary medicine to be used in conjunction with VEGF-A inhibitors for the treatment of wet age-related macular degeneration, which is wet AMD, and diabetic macular edema, which is DME. Opthea has completed a phase 1 out of 2A clinical trial in the US, investigating OPT-302 in wet AMD patients, and has 2 ongoing clinical trials still going on. Dr. Megan Baldwin was appointed CEO and Managing Director in February 2014. Dr. Baldwin brings 20 years of experience focusing on angiogenesis and therapeutic strategies for cancer and ophthalmic indications. Sorry, Megan, in advance. Prior to joining Opthea, she was employed at Genentech, the world leader in the field of angiogenesis-based therapies for cancer and other diseases. Over to you, Megan.
Thanks, Laura, and good morning, everyone. It's a pleasure to be here in this virtual setting to take you through an overview of Opthea's programs. As Laura said, we are an ophthalmology focused company developing an injection for the treatment of the 2 leading causes of blindness in both the elderly population but also in the diabetic population. And it's a pleasure to be able to have this opportunity to take you through our approach for developing that novel therapy. This is the requisite statement regarding forward-looking statements. Today, what I'm going to give you is a very brief overview of our technology at Opthea, a bit of a snapshot on who we are and the company itself. And then I'll really focus on giving you an overview of our clinical programs and the results that we've generated to date. We have completed now 2 clinical trials in our main focus area, which is the wet age-related macular degeneration space. This is the disease that affects the elderly population and is the leading cause of blindness in people over the age of 50. But we also have an ongoing clinical trial in diabetic macular edema, which I'll talk about in a little bit more detail. And this is our trial that we expect to report out results in June of this year. So in just a month or 2, those results are coming out. The company remains blinded or masked to those clinical results at this point in time. And then very briefly, as I round out today's presentation, I'll talk a little bit about our upcoming and near-term milestones and activities. So this is just a snapshot of the financial position of the company. We are trading now around about $2.30, $2.40, with a market capitalisation around about 640 million Australian dollars, or around $400 million US dollars. We've seen a very good response to our recent clinical trial data in wet AMD, and seen, as you can see there on the slide, a very nice response to that data in light of the fact that we've really generated positive clinical signals to improve visual acuity in patients with this disease, and achieved that outcome where a number of other pharmaceutical companies, many of whom are very large pharmaceutical companies, have failed to do so. So we're particularly excited about this technology, and you can see the market response to our therapy. We're excited to be moving forward into phase 3 clinical planning for that molecule as well, on the back of that data. Our shareholder register is largely comprised of institutional investors, many of whom have done a lot of due diligence on our programs. Those investors do understand the significance of the data that we've generated, the potential of our asset to address a very large unmet medical need in a space that is largely not as competitive as many of the other disease areas that you might be familiar with, cancer or immunotherapy, for example, being 2 key examples. There's really a very limited number of competitive products out there that may offer clinical benefit to patients with these eye diseases. And that's one of the reasons why I think we have a very large, high potential commercial asset on our hands. Our top 20 shareholders own about 75% of the company, largely institutionally held, and we're covered by the analysts, as you can see there at the bottom of the page. So just very broadly, our focus area is mainly on the wet age-related macular degeneration opportunity. This is a disease at the back of the eye, so it affects a disease tissue called the retinal tissue. This is the region of the eye that's required for highly detailed, sharp vision that is required for you to be able to read, to be able to drive a car, to be able to recognise faces, etc. very small impacts to visual acuity can actually have a profound impact on the quality of life for patients. And that's why even small benefits to visual acuity or vision in patients can translate into a very successful drug product itself, because vision is so critical to our everyday activities and our quality of life. So this disease is really driven by too many blood vessels growing at the back of the eye, and those vessels, when they are growing, becoming very, very leaky, and that fluid starting to accumulate at the back of the eye, which causes a swelling, it causes inflammation, and it causes cell and tissue damage that then affects your ability to see. In diabetic macular edema, many of the same processes occur, but they're induced, or they start because the patient has had a long-term hyperglycemic environment. So they're hyperglycemic, they've got diabetes, that triggers a series of pathways that lead to inflammation, it leads to fluid swelling at the back of the eye. And this is a complication of diabetes that then affects your vision. Our product is being developed for the vision loss associated with both of these disease processes. So let me tell you a little bit about how our drug works, and the pathway of molecules that I'm going to talk about are involved in making vessels grow, and they're involved in signaling and making vessels leak fluid. So you can see it's really at the central biology of both of those disease processes, which is where our molecule works. And what you're looking at here is a series of receptors that are primarily expressed on the surface of blood vessels. And the main driver of making vessels grow in this pathway is VEGF-A. This is a molecule that's targeted by a number of different agents that are currently approved, but all of those agents target the same molecule. So effectively, they share a common mechanism of action. They're all cannibalizing into the market share of each other. Eylea is another agent that also targets VEGF-A, but also targets two other members of the VEGF family. But basically all of these molecules are approved for wet AMD, in some cases also DME, in order to prevent blood vessels growing and to prevent their leakage. And they're marketed around the world by the companies which I've listed there. Our molecule is different. OPT-302 is a trap molecule. It's a protein that effectively mops up two alternative members of this pathway. And the important point here is that VEGF-C and D, similar to VEGF-A, drive processes through VEGF receptor 2, but those existing therapies don't actually block VEGF-C and D. So we need another molecule, and our molecule OPT-302 is a fantastic blocker, and it prevents the interaction with both of those receptors, and therefore effectively shuts down the other half of the pathway. Our proposal with this molecule is to use it as a combination product. So we're not seeking to replace any of these multi-billion-dollar drugs. We're seeking to add on to them and to tap into that market share. So we're not seeking to replace them. We don't need to be competitive with them. We just want to add on so that when a patient comes to the clinic, they receive 2 injections instead of 1, and therefore much more effectively block those pathways that cause the disease and cause the vision loss in both wet AMD and diabetic macular edema. And that's a little bit of a CSIRO generated molecular structure of our molecule. It's effectively a very potent blocker of VEGF-C and D, and structurally looks quite similar to Eylea. So this is a molecule that has a chemical structure that is not unusual, and physicians have a high level of confidence in using this kind of mode of therapy. It's currently a very large market opportunity in excess of $10 billion. Those drugs that I just pointed out to you on the previous slide generated revenues in excess of $10 billion in 2019. And as I said, that's really on the back of all competing for the same market share. We think that that market opportunity is completely available to new therapies coming on the market that offer additional benefit to patients. There are a lot of patients that don't respond optimally well to those existing therapies. Many of them have suboptimal visual responses, so there's still room to improve their visual acuity. And many of them continue to have fluid at the back of the eye, despite going on regular monthly treatment with one of those VEGF-A inhibitors. So that's an opportunity for new molecules that are coming through. These are diseases that are increasing in prevalence. The market opportunity is growing, and there's very few other molecules in development that may offer additional clinical benefit to patients. The endpoints that we use to monitor the efficacy or how well our drug works in these clinical trials are routine and non-invasive. We simply measure the ability for the patient to see at the start of the clinical trial and then compare that along the way up until the end of the clinical trial. And we're really looking for improvements or losses in visual acuity. Of course, with our molecule, we're hoping that the patient can see more letters on an eye chart after they've had their treatment. And we couple this with very sophisticated imaging techniques, again, that are non-invasive, that require just effectively a photograph of the back of the eye, but we can very specifically quantitate how much fluid is at the back of the eye. So our strategy with the molecule is to use it as a combination. We want to demonstrate better or superior vision gain in these patients. Right now, we're administering the drug on an every 4-week dosing cycle, but we have the capacity in future clinical studies to also look at less frequent dosing strategies, but to potentially also produce a co-formulation with other agents that are either approved or coming through in development, which would reduce the injection frequency and potentially co-administer a VEGF-A inhibitor with our VEGF-C/D inhibitor. And as I said, I've talked about the competitive landscape here being relatively sparse. Just as a snapshot of our clinical program, we have completed a 51-patient phase 1/2A clinical trial where we demonstrated that we met the clinical endpoint of that study, demonstrating safety. That led us into the very large randomised controlled phase 2B clinical trial, which was a global study, and I've got a number of slides in this deck to take you through that data. Effectively, that was a proof of concept study, a very large trial, even by global standards, one of the largest clinical studies that's been generated in phase 2B setting in wet AMD. And of course, we demonstrated that we hit the primary endpoint in that clinical trial. The company is now largely focused on taking that molecule forward into the final stage of clinical development, which is a phase 3 program that will be comprised of at least 2 concurrent phase 3 clinical trials, and to reading out our 3rd clinical trial, which is the diabetic macular edema study, which I'll also take you through today. We were very excited about the outcomes of the phase 1/2A study for a number of reasons, but one of them most particularly was that in that early clinical trial, when we injected OPT-302 on its own without a background standard of care, the patients did very, very well. And this was a very clear indication that OPT-302, when administered on its own, does improve visual acuity. And you can see here the number of letters. There was 5.6 letters on an eye chart, so just over a line gain of vision in patients. And that's really when it wasn't even administered with any of the other standard of care treatments. So we're very encouraged by that level of activity of OPT-302. And then both in combination in treatment naive as well as in prior treated patients, patients did very well relative to what we would expect as well. So that was a precursor for us undertaking this phase 2B clinical trial, 113 clinical sites across 10 countries, the US, 8 countries in the EU, as well as Israel, randomised controlled clinical trial, and a very robust clinical study design, which you can see here. We looked at 2 doses of OPT-302 in combination with the standard of care treatment, which was Lucentis, or also called ranibizumab. There was 2 doses of OPT-302 with that standard of care therapy. And we asked the question, does addition of OPT-302 to that standard of care treatment result in improved visual acuity in those patients compared to the control group that's shown there in grey? That control group only received Lucentis treatment. And so we're looking at the 2 drugs versus 1. Do our patients do better when they receive OPT-302? Each patient was dosed on a monthly basis for up to 6 months. And so our primary endpoint was at week 24, which is a 6-month time point, asking the question, what does their vision look like at that 6-month time point compared to baseline? And the answer to that is that we hit the primary endpoint, demonstrating superiority in visual acuity over Lucentis. We demonstrated a 3.4 letter gain, which hit a p-value of 0.0107, with 3.4 letters over and above what we saw in the control arm. Here now we're looking at 14 letters of visual acuity gain after the 6 months of treatment, which is approaching almost 3 lines of visual acuity benefit when it was administered in combination with Lucentis, and not as remarkable in the control arm. Clearly these patients did not do as well. The difference between the treatment groups was evident from 8 weeks onwards. And then that difference in the improved visual acuity was evident over time and throughout the entire course of the clinical trial up to the 6-month time point at week 24. So a visual acuity benefit, OPT-302, hitting the statistically powered endpoint, demonstrating a significant gain in visual acuity. This visual endpoint was also accompanied by anatomical changes, which also helped to validate the result. Changes at the anatomical structural level included a reduction in retinal thickness, which is what you're looking at here. We saw more fluid resolved and a less thick retina when patients received OPT-302 combination treatment compared to the control. When we looked at the percentage of patients that were actually dry at the end of the study, there was a high proportion of patients that had complete resolution of subretinal fluid as well as intraretinal fluid after the treatment on the combination study. So we're resolving fluid and we're improving vision. We are also improving the size of the lesion area itself. So we're actually reducing down the area over which the lesion is diagnosed. So there's a regression of lesion area, and that's concordant with a reduction in the number of vessels and the lesion area that those vessels actually occupy. So there's an effect at the blood vessels, an effect at the fluid level that's then translating into improvements in visual acuity. What is very important in this class of molecules is the safety profile. We're injecting this into the eye. It is very, very important that you have a very low level of inflammation through the injection process. And I'm very pleased to say now across a large number of clinical studies across multiple clinical sites that we have seen a consistent, well-tolerated safety profile of OPT-302. And this is particularly important given the very highly publicised concerns about safety with some of the other molecules that have been completing phase 3 clinical trials or actually even going into launch. Our intraocular inflammation levels are very, very low. So in conclusion, we're very happy that we've met the primary endpoint of this study, demonstrating superior gain of 3.4 letters, which was highly statistically significant. And it was against a control arm that really outperformed what we had expected as well. This was accompanied by improvements in the anatomy of the lesion at the back of the eye. We reduced the thickness of the retina and we reduced the amount of fluid at the back of the eye as well. And we reduced the overall lesion area. These are all very much supportive secondary endpoints that support that improvement in vision. What I haven't shown you today is some additional data analyses that we've done. We now understand population groups within the wet AMD disease that respond very, very well to OPT-302 treatment. We have a very good understanding of what kind of patient does exceptionally well. And in general, those patients that don't do so well on existing standard of care treatments tend to do better when they're on OPT-302 combination therapy. And this of course speaks to the fact that we hit a different part of the pathway, and that when we use our drug together with those standard of care treatments, we get a better outcome. So I think from a scientific perspective, how our data actually comes together all makes sense from a very strong scientific rationale perspective. And of course, it's in the profile of a very good safety and tolerability profile in combination or even as a monotherapy. I'm going to very briefly touch on our diabetic macular edema program right now. This is the trial that is a smaller clinical study. It is a phase 2A study, an investigational trial. It's the first study that we've read out or will read out in over 100 patients with diabetic macular edema. And it is a study that we've reported out the phase 1B component, but await the outcomes in the phase 2A component. Expect to report them.
Sorry, Megan, we seem to have lost you there for a little bit.
Standard.
Oh, and she's coming back in. We're having some technical difficulties here.
Patients do very, very well, but two patients don't have as profound visual acuity gains despite ongoing and very regular treatment.
Sorry, Megan, we were just having a bit of a technical hitch over here. We can hear you now. You were cutting out before. Maybe if you could return back to the slide that you were on and start from the beginning on that one.
Okay.
So if you could share your screen again, that would be great.
Okay, sorry, it's just dropped out.
Thanks, Megan.
Okay, are we back online?
We're back online.
Great, thank you. So I'm just going to highlight that we are targeting patients that have persistent DME in this trial. These are patients that have been on prior therapy, but they haven't done so well. They're effectively the patients that have received regular ongoing therapy. They're going back to their doctor, they're getting treatment, but their visual acuity is not improving to the extent that the physicians are happy with. We recruit those patients into our clinical trial, and just trying to circulate my screen now. Paradigm, how do they do? Do we improve visual acuity? The first part of this clinical trial was a dose escalation study. So we have to look at the safety profile as a low, medium, and then a high dose together with a standard of care treatment. In this case, in this trial, we used Eylea, again, a VEGF-A inhibitor, a different standard of care treatment to build up our knowledge of how we combine with different agents in this class of drugs. And we injected patients on a monthly basis for 3 months in this clinical study. Very briefly, the safety results were very, very clean. We didn't hit a maximum tolerated dose. You can see all of the zeros across the screen there. Really no safety concerns were highlighted in the first 9 patients treated in this dose escalation study. Very encouragingly, we saw dose-dependent improvements in visual acuity when patients were treated with OPT-302 and the standard of care treatment. And across the board, that was a 7.7 letter gain across the 9 patients. So patients tended to be doing very, very well, and particularly well in the 2 milligram dose group. With aflibercept, this is a small 9-patient study, but it is very encouraging to see dose responsiveness. When we increased the dose, we got a better effect. And this was concordant with a reduction in retinal fluid in those 9 patients as well over time. One point I want to talk about in terms of the context of the results of this clinical study was that there were 5 patients with diabetic macular edema that had DME in both eyes. Now, we are not able to inject our treatment drug into both eyes. What happens is that we recruit and involve only one of the patient's eyes in our clinical trial. The other eye, which we call the fellow. What we have now is a series of 5 patients that in the study eye received OPT-302 plus Eylea, and in the other eye only received standard of care treatment. And then we asked the question, how did the study eyes go? How did their visual acuity look relative to the fellow eyes that didn't receive OPT-302 after the 3 months of treatment? This is effectively a within patient control group. You can see here the response in the study eyes, which received OPT-302 plus Eylea, was 10 letters. That's 2 lines of visual gain on an eye chart, compared to the other eye in the same patients only getting 2.6 letters of visual acuity gain. And this was even in a more profound effect when we looked at retinal thickness, which was profoundly reduced in the study eyes that had OPT-302 treatment with standard of care, compared to the fellow eyes that only received the standard of care treatment. So this is very encouraging data, and we believe that that bodes well for the outcomes of our larger phase 2A clinical trial. In this phase 2A clinical trial, we're looking at the proportion of patients in the combination group that actually have a 5-letter or more gain in visual acuity. It's a responder analysis. We know what to expect with aflibercept, and we're looking for an improvement over what we expect in the treatment in combination with our drug. The comparison to the aflibercept control group comes in the secondary endpoints, where we'll be looking at the differences between the visual acuity responses between the groups. We'll also be looking at anatomical outcomes, similar to what we did in the wet AMD study. So a number of important secondary endpoints, but a responder analysis in our primary outcome measure for this clinical study. And as I said, we anticipate reporting that out in just a number of weeks' time in June of this year. We have not been overly affected by the COVID-19 pandemic situation with the finalisation of this clinical study, which we're very fortunate about, given that it was a study that involved several countries around the world. Safety profile to date from that phase 2A study continues to be very clean, very well tolerated, and no ocular serious adverse events have to date been reported. The majority of patients now are off study completely. All of them have completed the week 12 visit, which is required for the calculation of the primary endpoint. And this is a little bit of a summary of where we are overall in our clinical studies. You can see here, we've dosed over 1,500 doses of OPT302 across our phase 2B and phase 1/2A wet AMD studies, and nearly 300 patients with diabetic macular edema have been treated. So very happy with the safety experience we have with the molecule in an environment where some of the other molecules are seeing some safety signals or concerns. Our safety profile to date is very, very good with OPT302. As I mentioned earlier, we are planning our phase 3 program. Consider this as a framework on which our program will be based. It will be a 2 phase 3 clinical study program. They'll be conducted in parallel and concurrently. And we'll be doing OPT302 treatment at the 2 milligram dose level in combination with a VEGF-A inhibitor, compared to patients that only receive the VEGF-A inhibitor. Very similar, in fact, design to our phase 2B clinical trial. And we recognise that keeping things quite similar to phase 2B is favourable. And with such great results in the phase 2B, we believe that we have a very good phase 3 program coming through in the planning phases. We hope to be recruiting patients in our phase 3 program towards the end of this year. Upcoming milestones, we've had a very busy year following the positive readout of the phase 2B clinical trial. We're in a good, strong financial position, having raised 50 million Australian dollars in December of 2019. So that gets us well into the planning phases of phase 3. It gets us through the diabetic macular edema endpoint. We are not fully funded through the phase 3 clinical trials, as you can imagine, but we're some months away from doing that. And we have multiple options on the table regarding moving into phase 3. We've progressed our cGMP manufacturing. This is the standard of manufacturing you require in order to take this into phase 3 development and into commercialization, actually. And we've completed, obviously, patient dosing and patient recruitment in the 2A